Hello everyone, I'm Tarini. I work at Qubrid, and today I wanted to share the work we are doing on Quanta-Bind, developing a quantum computing pipeline for biologically relevant systems. This work is part of the Quantum for Bio grant, which is administered through the Wellcome Leap Foundation. And while the PI is Dr. Kanav Satya at Qubrid, we have a lot of collaborators across the board, both in academia and industry, including MIT, UChicago, North Carolina, NVIDIA, Wisconsin-Madison, and the Argonne National Lab. So it's a pretty broad collaboration. So today I want to talk about why we are developing this pipeline, the actual pipeline and its component methods, share one brief part of the optimization we are doing. We are using multiple methods, but in the 12 minutes I have, I want to focus on one of them and talk about some of the things we're going to do in the future. So first, motivation. Intrinsically disordered proteins, so proteins that move a lot, that don't necessarily have one fixed structure, interact with metals in the human body, in the brain, in the blood-brain barrier, and they're thought to play an important role in the pathogenesis of Alzheimer's disease. So here on the right, you have amyloid beta, and this is a massive protein. Misinteractions of these proteins can lead to diseases such as Alzheimer's. So by being able to run calculations at a much higher degree of accuracy using quantum computing methods, the question we are trying to answer is, can we eliminate or confirm any specific metal amyloid beta conformations? Can we improve our understanding of various Alzheimer's hypotheses? And can we study any drug-metal interactions to study off-target effects? Broadly speaking, the question we are trying to answer is, can we aid the drug design process by better understanding metal-protein interactions? And while we've focused fairly extensively on the amyloid beta protein, I'll share a little bit more about how this is a very generalizable pipeline. So amyloid beta, as I said, ranges from 36 to 43 residues. The 42 residue system, so these are amino acids, think of them as just like 42 components to the protein, is the most toxic. And that's what we are trying to do. Can we improve our mechanistic understanding of amyloid beta with metals in their human body, specifically in the brain? And on the right here, this is an iron-sulfur cluster, which is also supposed to be incredibly important to metabolic pathways in the body. So this is part of a broader work where we study biologically relevant systems with protein-metal interactions. So let me talk about the actual Quanta-Bind pipeline itself. We start here on the left, where you have some biologically relevant system. We fragment the protein with FMO, fragment molecular orbital method. This is a fully classical method. Next, we take classically intractable fragments. So these are fragments that are either too difficult, too large, or too strongly correlated to be run on classical computers, and we pass them further down the pipeline. Here we have two methods to treat the electronic structure problem of the system. One is quantum bootstrap embedding. The second is local active space VQE. We use GSE, which stands for generalized superfast encoding, to map the fermion problem to a qubit problem. And then we eventually run this on a quantum computer. So broadly, this is the pipeline. Let me talk about each method specifically. The FMO method is developed with Yuri Alexeyev, who's at NVIDIA. Basically, what we do is we break up a larger molecule into smaller fragments to make the problem more tractable. And we want to do this without losing any chemical or physical information in the system. So bonds are broken heterolytically, and a bond is moved with a proton. As an example, here's propanol, and you see that what we do is we break it up into roughly equally sized systems with 8, 8, 8, and then 10 electrons, respectively. So what we are doing right now is working with amyloid beta 16, a smaller version of the full protein. It's considered a minimal metal binding domain. And what I mean by that is it's all the parts of the protein that are involved in interacting with the metal. And it's only, not saying it's small, it's still over 250 atoms. It's only 16 amino acids. So we work with that, and using FMO, we split that up into 17 fragments. The smallest fragment is just a metal ion in the center. That could be zinc, that could be copper, that could be iron. And the largest fragment contains 94 electrons. we get some pretty good results with FMO, and we pass these fragments further down the pipeline. The next step in the pipeline is localized active space methods that are developed by Dr. Gagliardi's group at University of Chicago. So in case you're not familiar, what we do here is a hybrid quantum classical method that uses a last SCF, so localized active space, self-consistent field wave function for each fragment, and then we use VQE as a correlator between fragments. This method really shines where you have strongly correlated subunits because it recaptures a lot of the correlation energy that would otherwise be lost. Now, as you can see from this image here, you can use different methods as your inter-fragment correlator. So you could use ADAPT, you could use unitary coupled cluster, you could use USCC, which is unitary selective coupled cluster. And let me share just a little bit more about that. So last USCC, what we do there is we pre-select the most significant amplitudes from the original UCC ansatz, and then we define some threshold epsilon. We only keep all the amplitudes that are above that threshold. And what this does is that it significantly reduces the C0 gates needed by 85 to 90%. It wasn't working. Okay. And this helps increase algorithm efficiency and processing time because you have such a significant reduction in your ansatz size, in your circuit depth. Another option here is LAS ADAPT, where we use the ADAPT wave function combined with LAS onto the original UCC ansatz. This requires more VQE calls, but the overall ansatz is more compact, so it offers another option as a correlator between systems. We get some incredible results with this method, and in the interest of time, I just want to focus on one system here. So that system there is still being, and what's of interest is how the system isomerizes, so how the angle between the two ends of the molecule change as we change the angle from 0 to 180 degrees. And you see that LAS SCF, which is technically a fully classical method, does not capture a lot of the correlation energy. But then adding on LAS USCC, so adding in the quantum component to it, recaptures the correlation energy as seen with CAS, complete active space methods. Next, I want to talk about another component of the pipeline, quantum bootstrap embedding, which was developed by Professor Troy Van Voorhis at MIT. This method is analogous to classical bootstrap embedding, where originally you would have a system, you'd fragment it with overlapping fragments, you'd have some classical eigensolver, match one RDMs, and then generate the potential and go back. Repeat this self-consistently. With quantum bootstrap embedding, we are doing that, but slightly different. So we have a quantum eigensolver now, so that could be VQE, that could be QPE, and you're doing matching with RDMs, with swap tests and amplitude amplification. Then you look at the overall chemical potential of your system, the global chemical potential, use that to match and reconstruct your entire fragment energy. So with QBE as well, we get some really interesting results. And again, in the interest of time, I want to focus on one particular one, which is that as you improve your fragmentation, so BE1, BE2, etc., just refers to the degree of fragmentation. The number is the number of nearest neighbors you're including. And as you improve your fragmentation size, as you increase it, you see greater accuracy trends. Now, of course, that translates to increased circuit depths, increased measurement overhead, but it's an interesting result to show the accuracy trends as we increase our fragment sizes. The next part of the pipeline is generalized superfast encoding, which was developed by Dr. Setia. I'm sure everyone here is familiar with the Jordan-Wigner mapping. It's a classic. What you're doing there is mapping the occupation number of your orbitals to your qubit state. With GSE, this is a graph-based mapping. Orbitals are placed on vertices and then mapped to qubits on edges. So this is kind of what it looks like. You have your orbitals in green on vertices that you're mapping to the qubits that get mapped to the edges of this graph. This is done using edge operators, which are as follows over there. Now, GSE technically requires more qubits than Jordan-Wigner would, but it comes with error correction properties. Once the graph has weight 6, it can detect all single qubit errors, and it often offers lower circuit depths than Jordan-Wigner because of the locality of the Hamiltonian terms. So here's a quick example of GSE providing error correction properties, where you see that it gets closer to the unmitigated energy, the exact energy than unmitigated would have. Now, I want to talk about optimization, and there are many, many ways we can optimize this entire pipeline. We can optimize it globally across the whole pipeline. We can optimize each method individually. We can make approximations and show that they don't affect the accuracy of the system. What I'd like to talk about is one specific method called PI, so parallelized implementation of excitations. What we do here is we break up all of our excitations into singles, doubles, triples, so on, sort them by type, and then create an ordered ansatz instead of the original ansatz. What this allows us to do is excitations that are similar in nature, we end up with a lot of stacked C-naughts, and these C-naught ladders can be effectively canceled. This makes the overall circuit really efficient. So as an example, this is results with the Kramer's dimers. So this is a 2-chromium system. Those epsilons on the left there are epsilons we chose for the system for last USCC. Remember, that is a value of how many T amplitudes you've selected in your ansatz. With the original last wave function, you see the total operation circuit depths are quite high. After implementation and post-processing with Pi, we see roughly a 50% reduction in total operations and circuit depth. In the future, we want to continue various optimization techniques across the board. We want to continue works on error detection, error mitigation, and error correction, and we actually want to run this on quantum hardware. So, as I said, this is a massive project with many collaborators. A huge thanks to all of them and some of the PIs of the project. I'm happy to take any questions. Thank you.